Welcome to our deep dive today. We're going to be looking at some fascinating research on mitochondrial DNA mutations.
Yeah, in fruit fly.
In fruit flies, you got it.
We're going to be taking a look at what happens when these mutations just go kind of crazy.
Yeah.
And how researchers found a way to actually rescue the flies from some really serious consequences.
It's pretty wild. So we're working with excerpts from a scientific paper for this one, but don't worry, we're going to break it all down.
Yeah, we'll make it understandable.
Make it super interesting for you. And get this, the research uncovered a connection to something called autophagy.
Yeah.
Which basically acts like a recycling system.
Yeah, for your cells.
For your cells, exactly.
A crucial process.
Right.
But as we'll see, it's not always a good thing.
Not always a good thing. All right, so first things first. Why are we even focusing on mitochondrial DNA mutations? What makes them so important?
Well, you know how mitochondria are often referred to as the powerhouses of the cell?
I heard that.
They're responsible for generating the energy your cells need to function. Right.
But here's the thing. Their DNA is actually separate from the DNA in your cell's nucleus.
Oh, interesting.
And it's actually more susceptible to damage.
Oh, so any damage there has a big impact on the cell's energy production.
Exactly.
Makes sense.
And these mutations can be linked to a whole wide range of diseases.
Okay.
From rare genetic disorders to more common conditions like heart disease and even neurodegenerative diseases like Parkinson's.
That's a pretty broad spectrum of diseases.
Yes.
So how do fruit flies fit into all of this?
Fruit flies, or Drosophila melanogaster, are incredibly valuable for genetic research. They reproduce really quickly, they have a relatively simple genome,
okay,
and many of their genes and pathways actually function very similarly to those in humans.
So they're like a simplified model for understanding human genetics.
Exactly. In this study, researchers used a special type of fruit fly with a mutation in its mitochondrial DNA polymerase.
Okay.
This polymerase is like a spell checker for DNA, and without it, errors start piling up really quickly.
So without that spell checker,
those errors translate into problems for the flies.
Absolutely. These flies end up being smaller, their movements impaired.
Oh, wow.
And many of them don't even make it past the larval stage.
That's rough. Did the researchers notice anything else going on at a cellular level?
Yes. They observed reduced activity in several key mitochondrial enzyme complexes.
Okay.
These complexes are crucial for energy production.
Right.
So their reduced activity further highlights the dysfunction caused by the mutations.
Yeah.
There was also a decrease in mitochondrial membrane potential.
Okay.
Basically, the energy charge of the mitochondria was lower.
Got it.
And they found reduced activity in an enzyme called aconitase,
Okay.
which is sensitive to cellular stress. So all of this is pointing to some serious problems in these flies.
Wow. It sounds like these flies were in really bad shape.
Yeah, they weren't doing so well.
But I thought you mentioned something about a surprising solution. What did the researchers find?
Well, they performed what's called a genetic screen, where they tested different genes to see if any of them could improve the fly's survival.
So they were looking for a genetic fix, so to speak.
Yeah, and what they discovered was completely unexpected.
Oh.
Reducing the expression of certain genes actually allowed the mutant flies to live into adulthood.
Wait, reducing gene expression?
Yeah.
Isn't the usual approach to add or fix genes, not reduce their activity?
That's the typical line of thinking. But in this case, less was definitely more.
Less is more.
Yeah, it seems counterintuitive, but remember, these flies are dealing with a process that's gone into overdrive.
Right.
By dialing it down, they were able to restore some balance.
Okay, so dialing things down was the key. So which genes were involved?
There were several, and they're involved in seemingly unrelated processes.
Okay.
Some are connected to autophagy, the cell's recycling system, while others are linked to mitochondrial protein import,
Okay.
insulin signaling, and even nutrient sensing.
Wow, that's quite a mix. Insulin signaling and nutrient sensing sound pretty far removed from cellular cleanup. How do those tie into the mitochondrial dysfunction?
It's fascinating, right? All of these pathways are actually interconnected.
Okay.
They all play a role in how cells respond to stress and how they kind of maintain their energy balance.
So it's like a complex web, and by adjusting one part of the web, they were able to influence the entire system.
Precisely. When a cell senses a problem, like these mitochondrial mutations, it triggers a variety of responses to try and compensate.
Okay.
One of those responses is ramping up autophagy to try and clean up the damage.
That makes sense. But as we saw, too much cleaning can actually be harmful.
Right, and that's where those other pathways like insulin signaling and nutrient sensing come in.
Okay.
They influence how cells utilize energy and allocate resources.
Okay.
By adjusting these pathways, the researchers essentially help the flies optimize their energy usage despite the mitochondrial dysfunction.
So they helped the flies find a workaround for their faulty power generators by improving the efficiency of the rest of the cellular machinery.
That's a great way to put it. And here's where things get really interesting. This research isn't just about flies.
Are you saying what works for fruit flies could potentially work for humans too?
It's certainly possible. The genes and pathways involved in this study are very similar in humans, which is why fruit flies are such valuable models for research.
Okay, but how could this research actually help us understand human diseases in aging?
Well, this study sheds light on the role of excessive autophagy in cellular dysfunction.
Oh,
Scientists already knew that autophagy is important for cellular health,
right.
but this research suggests that too much of it can actually be detrimental, especially when it comes to mitochondrial diseases.
So if we could find ways to target and control autophagy in humans, we might be able to develop new therapies for these diseases.
Exactly. This study provides a starting point for exploring those possibilities. Imagine developing drugs that could fine-tune autophagy, preventing it from going overboard and causing harm.
That would be incredible. What about aging? How does this research connect to that?
Well, remember how we talked about mitochondrial DNA mutations being linked to aging?
Yeah.
This study suggests that it's not just the mutations themselves that cause problems, but how the cells respond to them.
So it's not just about the damage itself,
Yeah.
but how our bodies attempt to repair it.
Exactly. And if excessive autophagy is part of that problematic response, finding ways to regulate it could have implications for healthy aging.
That's pretty amazing. Tiny flies helping us unlock big secrets about human health and longevity. Did the researchers happen to pinpoint any specific parts of the autophagy process that seem particularly important for rescuing the flies?
That's a great question, and yes, they did. They found that targeting the nucleation process of autophagy was especially effective.
The nucleation process. Can you remind us what that is again in the context of autophagy?
Of course. Think of autophagy like a construction project. Nucleation is the very first step, where the foundation for the cellular cleanup crew is laid.
Oh,
It's like gathering the building materials and setting up the work site.
So by targeting this early stage, they were able to prevent the entire cleanup project from spiraling out of control.
Exactly. And that's a crucial finding because it suggests that we might not need to completely shut down autophagy to see benefits.
Okay.
We could potentially achieve the desired effects by simply tweaking specific steps in the process.
That sounds like a much more targeted and potentially safer approach. So what does this all mean? Where do we go from here?
This study opens up a whole new world of possibilities for research and drug development.
Okay.
We have a better understanding of the role of excessive autophagy in mitochondrial diseases and potentially aging. And we have a potential target, the nucleation process, for developing new therapies.
Right.
Of course, more research is needed to translate these findings from flies to humans, but it's a very promising start.
It definitely is. And I have a feeling that the next time I see a fruit fly buzzing around, I'm going to look at it with a newfound appreciation for the scientific contributions these tiny creatures are making. But before we wrap up, is there anything else about this study that you found particularly surprising or intriguing?
What really stood out to me was that even though most of the rescued flies still had a lot of those mitochondrial DNA mutations, they were able to survive.
Interesting.
This really emphasizes the idea that it's not just the mutations themselves, but the cell's response to those mutations that determines the outcome.
Yeah.
It's a powerful reminder that our bodies have this incredible ability to adapt and compensate.
Yeah.
And that sometimes a little less intervention can go a long way.
I love that. Sometimes it's better to work with our body's natural processes than try to completely override them. It really highlights the complexity and interconnectedness of these cellular systems.
Yeah, it's a delicate balance, and this research gives us a whole new perspective on how to approach it.
Absolutely. And it speaks to the power of just basic research, you know?
These unexpected discoveries can open up entirely new avenues for treating and preventing diseases.
Yeah, it's amazing to think that such tiny creatures can contribute so much to our understanding of human health. So next time you see a fruit fly buzzing around, maybe give it a little nod of appreciation.
I completely agree. They're helping us unravel some truly profound mysteries.
For our listeners who want to delve deeper into this research, we'll be sure to include links to the original study in our show notes.
We encourage you to check it out.
But before we wrap things up, I want to leave you with something to ponder. We've been discussing how preventing excessive autophagy rescued these flies, but what about the flip side? Could there be situations where boosting autophagy in a controlled and targeted manner might actually